in biology class, you probably called this the endomembrane system. Should be kind of a review, it's not a big deal. It's just the way proteins are secreted from a cell. Inside the nucleus, messenger RNA is produced. Messenger RNA goes out to the cytoplasm where there are ribosomes. With regard to hormones, these ribosomes are going to be on rough ER because we need to modify this, package it for transport out of the cell. So ribosomes on the rough ER would make this immature hormone here. They would then package it into a vesicle, which would go to the Golgi. The Golgi would then modify and more, package it into another vesicle, which would then release it by exocytosis out into the cytoplasm, or into the extracellular fluid. And then it would get absorbed through the filtration slits in the capillaries into the blood plasma, and then it's transported out to its target. Right. So this is just nothing more than the endomembrane system working. This is actually the way anything that is exported from a cell is produced and packaged, any protein. Right. If this were a steroid hormone, the difference would be that instead of rough ER, we would have smooth ER. We probably wouldn't package it in any kind of vesicle. As the smooth ER made it, it would just dissolve through the membranes straight out into the cytoplasm, or to the extracellular fluid out here. Okay. But because this is hydrophilic, we need to package it, or we can package it in these vesicles for transport. Okay. And again, what the hormone is doesn't matter. If it's a protein-based hormone or a peptide-based hormone, it's going to get produced that way. There's an example of the activation of insulin. As I mentioned before, you have to cleave a piece out of the primary transcript. This is the first thing that's made from gene expression. You'll notice there's a little bit of modification here. We call these disulfide bridges. They just give us some secondary structure, right? And then to make it active, you have to cleave out this portion that's called a C-peptide or C-protein. And this is the functional part of it. What's this? It's just waste. And not really, but we'll say it is. And this is what then regulates its target cells if there is a receptor and makes those target cells increase the absorption of glucose. Again, just modification. Nothing special there. Again, just to review, how does that peptide hormone work? Remember we said that hydrophilic signals bind to surface receptors. Those surface receptors can be linked to an ion channel, in which case if it binds to the ion channel, the channel opens and closes and you get ion flux. What ions move depend upon what the channel is, right? Because that channel is specific. That membrane receptor could be linked to an enzyme that will be activated upon binding with the signal and will trigger some response. A common example is what we call a TK receptor. TK receptors are receptors linked to tyrosine kinase. In the presence of the signal, tyrosine kinase is active, sticks phosphates on tyrosine residues, activates proteins inside the cell. Very common, very common. But many are linked to second messenger systems. And the second messenger system, remember, you have a receptor linked to GPCR, G protein coupled receptor. You have a receptor linked to a G protein. When the receptor binds the signal, the G protein is activated. That activates another enzyme in the cell. Which enzyme it activates will determine which second messenger you produce. Adenylyl cyclase is part of the system where you get cyclic AMP. Phospholipase C is part of the system where you get diacylglycerol, right? And then those second messengers that you get will trigger some response. And we saw that most of them, both cyclic AMP and diacylglycerol, they work by activating other kinases in the cell that phosphorylate proteins and turn proteins on. Which one of these? What are we talking about?